for ABL Bio. ABL Bio is a pure antibody company and it works on cancer immunotherapy as well as degenerative brain disease. The presentation will be done by President Lee Sang-eun of ABL Bio. Thank you for coming. President Lee, welcome. I heard you've prepared a lot for today's presentation. Please begin.
Sure. ABL Bio is located in Pangyo. It was founded in February 2016 and went public on Costock in December 2018. We have about 85 staff members. And ABL Bio stands for Medicine for a Better Life. So ABL is an abbreviation for a better life. So literally, we wanted to provide a better life for patients. And I myself is CEO of this company. And compared to other companies, we have special experts like this. There are 6 board members supporting me. And just like mentioned before, our company develops dual antibodies. And this slide looks a little bit sophisticated, but we wanted to develop a innovative agent. And we learned that we need a strong platform, because if we have a strong platform, then even if we may fail in some parts, we can overcome with that with other agents. So we especially focused on developing this platform. And in terms of this dual antibody technology, while we rapidly grow, we wanted to take similar steps with the global trend as well. And just like mentioned before, we have 2 different approaches to this technology. One is cancer immunotherapy, and the other one is CNS, the degenerative brain disease. We developed this GrabBody platform, which I'll dive into that in some few minutes. And there's this platform called GrabBody T and GrabBody I. So we wanted to develop these new platforms to come up with dual antibody therapy. And I'll later discuss ABL001. We have just finished our first clinical trial, and we will soon begin our combined treatment 1B clinical trial. Next slide. There are only 5 to 6 platforms. However, each has about 3 to 6 subcategories. We have recently finished our phase 1 ABL001, and the green colored text is currently we are working a partnership with Revocam, which is another enlisted company on Costock for antibody drug conjugate. And the next one, immuno-oncology, GrabBody T and GrabBody TI. GrabBody T stands for T cell. So we work on this GrabBody T immunosystem. And next is GrabBody 100 series. So we're currently working on different technology partnership, and we're co-developing with Trigger overseas. And next one is we are working with AMAP, which is a NASDAQ enlisted company located in China. And next one is GrabBody B. B stands for brain, so we're working on Parkinson's disease as well as dementia itself. Next, before I dive into 001, I would like to mention angiogenesis. So in terms of anti-angiogenic therapy, there is Avastin developed by Genentech and Roche. They sell about 7 trillion Korean won. And next one is Lilly's, which sell about 1 trillion Korean won. And the problem of these agents is that they grow tolerance, but we wanted to develop a Avastin targeting UGF genes, and we wanted to suppress those DLL4. So this is a non-clinical data. We conducted animal trials, and as you can see in the middle graph, the yellow colored one, we have this high efficacy, and we compared it with Avastin and DLL4. So instead of this single administration, we learned that the combined treatment is better. And we began our clinical trials, and over 1.5 years, we went through from cohort 1 to 9. And what really is important about clinical trial is to learn if there's any toxicity if we administer this to patients, and if there's any toxic side effects. And the phase 1, the first clinical trial, is from a very low dose to high dose. High dose means 15 milligrams per kilogram. And the best result may be conducting a study on 2 different confirmed patients, which is called PR. For example, if you say the tumor size unit of a patient is 100, then it reduced by 30%, which became 70, which is a very good result. And in the future, continuously, we want to work on chemotherapy or PD-1 or Keytruda. We are planning to work on combined treatment, and we had discussion with FDA. And later, like after April or March, we are planning to go through phase one clinical trial as a combined treatment. And it has been some years since the beginning of this clinical trial, and as went by, we are seeing more fruitful results, so we're expecting to see even better results. Next is our non-clinical data in terms of cancer immunotherapy. Please take a look at on the left one. This PD-1 is very famous. For example, President Jimmy Carter got injected by Keytruda, which is a cancer anti-cancer, and he overcame cancer. This story is very famous. So a lot of patients are through administration of PD-1, but however, only 20% of patients show reaction. So a lot of global pharmaceutical companies are working on this combined treatment with PD-1 or working on finding dual antibodies for this. So on the right, it looks very sophisticated, but to put it simple, we want to enhance the Keytruda agent to make it even better. So we are mainly focusing on this called 41BB, which is a gene cancer immunotherapy. If you take a look at the picture on the left, it looks really sophisticated. So I cannot dive into details for bi people. However, if you administer with this, then it can boost your cancer therapy. So we will come up with dual antibody to overcome this. And if you take a look at this Bristol Myers, there is an agent called urelumab. So I circled it with a green circle. And this is very famous. A very famous company in the US developed this. BMS developed it. However, because of liver toxicity, they had to shut down the clinical trial. In order to overcome the side effects, we wanted to develop this T grab body I platform, and I'll explain about grab body T. So basically, in order to induce T cells, the location of dual antibody is really important. That is what we thought. And if you take a look at this yellow colored image, the circle image, the red and yellow colored ones are those cancer cells and anti-cancer cells, and the green one on the bottom is T cell inducing antibodies. So we attached two different antibodies, and by using this platform, we wanted to target the tumor cells well, and by activating the tumor cells, so it can kill those cancer cells. As shown on the right, what we can differentiate it from other companies is that these antibody, to which part of the antigen this antibody attaches is really important. And urelumab, they attached on the upper part, and Pfizer attached it in the middle, and our ones, they're to be found on the bottom. And in terms of having enough distance, and in terms of binding of antigen and antibody was really important. And we conducted a lot of animal tests, and we conducted various research. And as mentioned, there was cancer targeting agent, and we attached 41BB on the bottom. And as shown from the second image, we had 5 nanometers of distance from the dual antibodies. The activation level was really low. But if we attached cancer on the left and attached this antibody on the right, then it showed this kind of graph. And when we had more distance like this, then it showed much better activation level. So this grab body T is the one that we developed, and we're working with Yuhan Pharmaceuticals, Dongha Pharmaceuticals, as well as Trigger. And we conducted this mice study, and they overcame cancer. And there is this test called tumor rechallenging, which is very important in terms of science. We administered 6 different doses in 6 different times to get rid of cancer, and then we get rid of the anti-cancer. Then it's likely that they will grow cancer again. However, as a lot of time passed by, they didn't get cancer again, which means the grab body T is in terms of T cell activation. We not only do we reactivate this the generation, but we can come up with memory T cell. The term memory here refers to memorizing, the remembering the cancer cells. So when another cancer cell comes, you can kill this cancer cells. So we have found this mechanism, and based on this, we have come up with GrabBody T. And let's move on to GrabBody I. This is a combination of two different antibodies. So last week we had this ABL 501 project selected by Ministry of Health and Welfare as a pharmaceutical project, and we are preparing to go through a clinical trial in the United States, approved by the states, and we're working on this I platform. And in terms of GrabBody I, we are working on PDL1 and LAG3 suppressing dual antibody. And LAG3, as I have mentioned before, if you take a look at the image here, we have this dendritic cell and T cell. It activates the T cell pretty much. What better? So, based on the Keytruda, the conventional mechanism, if we combine this dual antibody on it, and we come up with dual antibody, and Roche, Zenko, or other famous companies are working on their first clinical trial, and for us, ABL Bio, early in next year, we are planning to begin our clinical trial. And in terms of M10404, this is a PDL41BB, and I think this is the best in class, and the mechanism, as I have mentioned before, Keytruda or Optivo or other PDO therapy, we can overcome with those side effects. And currently, we're in a partnership with Wuxi, which is located in China. We are producing this, and we have patents. And later in December, we are planning to go through IND filing to go, which is an application submission to go through a clinical trial, so we can administer these agents into patients. And we are almost done with monkey animal trial, and once completed production with from Ushi, we will begin developing our clinical trial. It's very soon. Let's move on to the next slide, which is ABL 503. We wanted to see the efficacy of this. So we conducted two different tests. It showed reaction to PD1L1, and for PD1L1 showing high efficiency and reaction, it showed good effects for those intermediate or high expressions. So when we planned this, we spent time agonizing, and we learned that patients showing at least intermediate level or high level would show good reaction for dual antibody. And let's move on to ABL 111. ABL 111 has to do something with Claudin 18.2, which is a very important agent. And this works on gastric cancer and pancreatic cancer. Especially for pancreatic cancer, there's no clear treatment. So by attaching this dual antibody, I personally think this could be a first in class. So by developing a dual antibody, we are planning to begin our clinical trial early next year. So likewise, we are working with Wuxi, the CMO company located in Shanghai, China. We are working on clinical samples production, and also early February next year, we are planning to go IND filing. Why is this attaching technology really important? Because in the circumstances where this micro cancer cells grow, T cell generation is really important. And to put it simple, when we conducted this tumor study, as shown in the red dots, T cells were actively generated from this antibody, which means tumor recruited, tumor followed T cells. Through this micro killing of cancer cells, we can cure cancer. And regarding that, we have conducted numerous animal studies. And surprisingly, when we conducted this placebo control, we saw this very high efficacy. If you take a look at the red graph there, we successfully decreased the size of tumor cells. Next one is ABL301. I would like to stress this one. And when we compare this to other global pharmaceutical companies, this is a very outstanding one. It looks very peculiar because The upper part looks very similar to other parts, but on the bottom, unlike other gravity B or among other ones, there is only one attached on the bottom. This one is called gravity B. B stands for brain, so there is a barrier called blood-brain barrier inside of the blood vessel of the brain, and we named this after brain to name it gravity B. And as shown on the slide, if you take a look in the brain, there are a lot of tight barriers in the brain called BBB. It's really difficult to penetrate this, so only 0.1% to 0.2% of administered drug can infiltrate the brain. So developing this effective platform to penetrate the brain barrier is really important. And as you can see from here, there are new targets, and by identifying them, and in BBB, we conducted this study to see good efficacy. And based on that study, we developed antibodies, and through a massive research, we could come up with the first generation one, and we completed this grab body B last year. And this first generation grab body, as shown here, it is symmetric one. But as time passed by, based on our research, we learned that this asymmetric one better infiltrates the brain barrier. So we came up with asymmetric one, and we developed this dual antibody platform. To put it simple, in terms of grab body, when we developed this asymmetric one compared to symmetric one, more dosage could go through infiltrate the brain barrier. So based on this in vitro and in vivo experiments, we could see six-fold to seven-fold of penetration. And in order to prove that, we have conducted animal research. We conducted this IV injection, and as time went by, we studied this, monitored this, and we wanted to see how much dosage could infiltrate the human brain barrier. And we wanted to see if we could actually see sevenfold or tenfold of infiltration as time passed by. We monitored this, and based on each time point, we monitored this for about 7 days, and we extracted brain parts from animals, and we calculated and as assessed. And for those a single antibody, it increased but decreased in 3 days. But for dual antibody, it increased, and then even if it decreased, it still is very high compared to single antibody. So compared to single antibody, this dual antibody, we monitored the dosage density, and it showed about tenfold compared to single antibody. And another notable thing is that when we increased the dosage of dual antibody, it continuously infiltrated the human blood-brain barrier and animal brain. So we increased from small dosage to intermediate level to high dosage. However, for dual antibody, it drastically increased the dosage infiltrating to the brain. And based on this study, we completed this GravBody B platform, and based on this research, compared to Roche or Genentech or Denali, among other competing companies, they when they developed their infiltrating platform and working on developing it. And if you take a look at the Roche's dual antibody compared to ours, well, the shape itself is really similar because it's also asymmetric, but ours has longer half-life. So if you take a look at the Roche's one, this red one is single antibody and the green one is dual antibody. The half-life is much shorter, which means the dosage infiltrating to the brain is decreasing. But for us, the half-life is almost the same, which means much more dosage can be delivered to the human brain. So we have developed this platform. So in terms of this platform, why are we stressing this too much? Is because this platform delivering to the human brain, we are working on this, so we can targeting the Parkinson's disease. We're working on the ABL 301. We're planning to go through a clinical trial next year, so we're working on monkeys clinical trials. So Alzheimer's disease can be covered by ABL 303 and ABL 304. The tumors in the brain, if we successfully infiltrate more dosage in the brain, then probably the cancer immunotherapy efficacy can increase. So that is our theory. And in terms of brain disease, probably there are a lot of enzyme related diseases. So if we attach enzyme to successfully deliver the dosage to the brain, probably we could see more fruitful results. So we can administer this to various diseases. When we came up with 301 for the Parkinson's disease, in terms of degenerative diseases, we wanted to see that, so we conducted this mice test. So we put mice on this treadmill, and it kept spinning, and mice kept running to stay on it, and we counted the seconds. Those healthy mice stayed up there for 200 seconds, and when there was a problem with this targeted dosage, the seconds decreased to 130 to 140 seconds, which is almost half. But when we administered this dual antibody, then we slowed down the decreasing speed, which was over 200 seconds. So this 301 targeting the degenerative brain disease, if we successfully infiltrate more dosage into the brain, then it increased the performance. So it opened a door to decrease and mitigate those serious illnesses thanks to the animal clinical trials. And we're working on this clinical trial to further develop this. And also, we tested rats, and there was no toxicity issue occurred. So, thanks to the animal clinical trial, we are 301 not only for rats, but we just completed our monkey tests, and there was no toxicity issue. So in the future, we'll keep working on developing this. The strategy by ABL Bio is like this. So just like I said, the grab body T cell activation and grab body I, and I stands for immunology. So this is cancer immunotherapy, and the next one is grab body B. Based on these three platforms, we will complete our platforms, and based on our excellent human forces, workforces, and strategies and technology development, we will continuously develop our pipelines. And as shown on the bottom, we have various open innovations, as not only in Korea but also globally. Take a look at this DKFZ, which is an organization, very famous organization, research organization located in Germany. And we're through this open innovation, we will cover those lacking capacity. And from non-clinical trial to clinical trial, we'll keep seek partnership with other organizations to develop innovative drugs. So as a dual antibody company, we will further develop and the 001 in order to continuously achieve not only 001 but other additional two new drugs and also other candidate agents. We will continuously work on our clinical trials to come up with additional agents in the future. Thank you.
Thank you. It was a presentation by Lee Sang-hoon. Thank you for your passionate presentation. Thank you.